Hello everyone. My name is Abhishek and today we are discuss about the microscope. Its title name is Statistical Analysis of Test Drug and Reference Drug Using Bioequivalence and Population Bioequivalence. So our introduction is bioequivalence B studies are connected to compare a tested drug and with a reference drug to ensure similar rate and extent of absorption using pharmacokinetic parameters such as Cmax, AUCT, and AUC infinity. This is essential for approval of generic drug as an ensure therapeutic equivalence without the need of the extensive clinical trial. Traditional approaches like average bioequivalence ABE focuses on comparing mean responses using statistical methods, but they do not account for variability among subjects. To address this population bioequivalence PBE, consider both mean and differences variability, which is essentially important for highly variable drugs. Viability BA from the foundation of the BE, describing how fast and how much drug reaches systemic circulation. The parameters such as Cmax, AUC, and Tmax help evaluate drug safety, efficacy, and absorption rate. Bioequivalence is established when the 90% confidence interval lies between the separate acceptable range of 80% to 125%. The main aim of this study is to assess whether the tested drug is equivalent to the reference drug in terms of both average performance and variability, ensuring safety, efficacy, and interchangeability. Here we discuss about the data. For BIB, here we can see the research paper link and data from 2 cross 2 crossover viability study on 16 subject. Each test subject received both test and reference with concentration measure from 0 to 16 hours, and AUC and Cmax were calculated. Bioequivalence assessed using log transform ANO. And secondary we are seen PBE data researcher paper link where in vitro data of test and reference across three stages. First is calculates D50 and span for drug release. Second is estimate variability. And third is analyze using PBE and with the upper confidence bound H. Here we are discuss about the abstract. This study compare a test drug and reference drug using average bioequivalence ABE and population bioequivalence PBE and methods based on 2 cross 2 crossover study with 16 subjects. Pharmacokinetic parameters Cmax, AUC 0 to T and AUC 0 to infinity were analyzed using log transform ANOVA and 90% confidence interval. Results show that Cmax and AUC 0 to T fall outside the acceptable 80% to 125% ranges, indicating lack of average bioequivalence, while AUC 0 to infinity meets the criteria. High variability was observed from Cmax and moderate variability for AUC. PBE analysis indicates failure to meet overall bioequivalence due to variability, although median response meets the requirement. Batch-wise analysis also shows inconsistency across stages. Overall, the test drug does not demonstrate complete bioequivalence and cannot be considered fully interchangeable with the reference objective. There are our five objectives. First is to demonstrate bioequivalence between the test and reference drug according to the regulatory guidelines. Second objective is to evaluate a bioequivalence between test and reference drug formulation by performing statistical analysis of log transform pharmacokinetic parameter using mixed effect model. Third one is to evaluate intra-subject and inter-subject variability of pharmacokinetic parameter to assess the and fourth is to assess the effect of sequence period and treatment. And last is the to compare the statistical performance of the test drug and perform drug using reference drug using population bioequivalence PBE methodology. Here we are see that parameter estimation. This table shows that we are estimate the parameter just like Cmax, Tmax, AUC 0 to T, AUC 0 to infinity, AUC lambda, etc. And the information about the parameter. Here are we seen the seven parameters. First is Cmax, and here we see the formulas of Cmax and definition. Similar for the Tmax, AUC 0 to T, elimination rate constant, lambda Z, Our fifth is a half-life lambda T 1 by 2 AUC 0 to infinity and LLOQ and LLQCT. Okay. So after we see why we go to B to PB. Population bioequivalence PB is essentially useful for highly variable drugs because such drugs show large variability sigma square in their response. Traditional bioequivalence B focuses on the mean difference between test and reference products may incorrectly reject the acceptable product when variability is high. PB overcome this limitation by considering both the mean variability expressed as this formula you can see, thus providing a more reliable assessment of drug performance across the entire population. Hence, PB ensures consistent efficacy and safety even in the presence of the high variability. And this is our population bioequivalence decision criteria. Population B criteria is here in formula. Then this is the parameters definition and linearized criterion for testing. Yeah, data values have been taken, and final decision rule is the 95% upper confidence bond HN less than equal to less than equal to 0 is a bioequivalent, and when HN greater than 0 not bioequivalence. Then we can discuss about the D50 and span in a practical size distribution. Then D50 represents the median particle size and help understand typical drug behavior. Span shows particle size variability indicating uniformity. Both are important for drug performance affecting dissolution, viability, and the formulation quality. Then particle size distribution PSD. After D50 median diameter, that is the interpretation. D50 is the particle size at which 50% of particles are smaller and 50% are larger. Then example, if D is equal to 5 mu m, half the particle are below 5 mu m and half above. Span measures the width spread of particle size distribution. Formula span is equal to D90 minus D10 divided D50. D90 is our small, small, smaller than this size, and D10 is the 10% particle are smaller than this size, and D50 is median particle size. And this is the our graph is shown in this poster and interpretation. When our span is lower, then more uniform particle sizes. When our span is higher, wider variation in particle sizes. There, we are going to our overall conclusion. In our first objective, uh, where test and reference drug is not bioequivalence according to the regulatory guidelines. Here is proof. The second objective is shows that when we are uh, when test and reference formulation do not meet the overall bioequivalence requirement when performed under log transform parameter using mixed sequence model. Overall the study shows that Cmax has a high variability, which means that the peak drug concentration changes are not within the same subject for AUC 0 to T and AUC 0 to infinity. The variability is moderate, which shows better consistency between test and reference. Between different subject, our variability is also moderate for all parameters. So overall, there is moderate to high variability, especially for Cmax. ANO analysis shows that sequence period and treatment effects were not significantly significant. P is greater than 0.05, indicating no carryover effect, no period information, and sorry, in influence and no significant difference between test and reference formulation. Thus, the crossover design is valid and appropriate for bioequivalence assessment. And our last and not least, the fifth objective is says that although the test and reference drugs show similar median response D50, they differ significantly in terms of overall performance and the variability span. Since PBE consider both mean and variability, the failure in variability leads to the conclusion that the formulations are not population bioequivalent. And there is our three tools, statistical softwares where here we are used in this our project. So first is Excel, second is R, and third is SAS program. And this is our image of the test truck and the reference for PB and B analysis. Okay. So thank you.